Wanted to try to have a conversation with you and also the other members of our DIPG collaborative with regards to your grant and to get a little bit more information about it. You're dealing with Rob from Isabella Marcus Foundation. We're dealing with Kim from the Robert Connor Dawes Foundation. We're also joined by Eleni of the Cure Starts Now Australia. With that in mind, I wanted to ask you specifically about this grant. This is a grant that's a very big grant. It's a very big project. You approached us about this, if I remember, about 2 years ago. Is that correct?
Yeah.
This grant also is primarily a medulloblastoma grant. And when it was put in for consideration to the DIPG collaborative, obviously that concentration is really about DIPG. But one of the things that are really core to our organization is that we want to focus on this idea of the universal cures and how it applies to other brain cancers and really what we can learn from these cancers that may in fact help all cancers, including DIPG and so forth. Can you talk a little bit about what this grant is and then also tell us why you think that it has those applications?
Sure. So this grant, I guess, started with the idea that there's parts of the brain microenvironment that seem to be important in terms of the growth of the cancer, but more importantly, in terms of how they respond to drugs. And what we realized when we started, so looking at our medulloblastoma models, when we were treating them with different drugs, we found that a lot of these processes and parts of the brain microenvironment, so the vasculature, the blood-brain barrier, and the matrix were changing in response to the drugs. And potentially when they relapsed, we saw that they were quite important in that setting. So what we found at that point was that, and what I found looking, there was no way to analyze that. There was no way for me to be able to ascertain what the impact of those things were on the growth of that tumor cell, and were they actually causing them to be more aggressive? How did that block our response to therapy? So the first thing that we needed to do, and really what started with us getting together, was to be able to establish new models to be able to address those two parts and how they interact with tumor cells. So look at the interaction between the vasculature and tumor cells. Number one, how does that impact the drug actually being delivered to a brain, any brain tumor? But two, how that interaction between the tumor cell back to the vasculature and what's happening while we're treating, because we know that that's changing from our data so far. In addition to that, and that's where Anna comes in. Anna's a brain vasculature expert, in particular in stroke and other brain type diseases where the blood vessels are really important. In addition to that, a lot of the resistance of another drug I was investigating, not the one in this application, I was seeing a lot of matrix changes in terms of the brain matrix, which is really, really unique compared to anywhere else in the body. And that's where Sam comes in. Sam is a melanoma researcher, but specializing on brain metastasis. So Sam works with chemists to try and rebuild the brain in a dish in that matrix and looks live at the interactions between the matrix and the tumor cells, because we know it's not just the soup that they sit in, but they actually talk to one another and are really important. At least we know from GBM in adults now and melanoma in brain mets, they're really important to the biology of that cell. So I thought, instead of me just looking at tumor cells when I treat them with my drug and looking at what happens in a mouse, which I really can't see so much, if I can model all those components together, I can look at all three. And that's really what this grant's about. And trying to basically show that all of these aspects influence the mechanical response of that cell, and that drives resistance. So that would be the main outcome, that we could show that it's a really new field of biology, and that's important to pediatric brain cancer because it's never been discovered. And then what can we do about it? Because there's drugs out there that are approved that can actually target that. So if that is a whole new biology, then essentially it opens up not only for us understanding about the tumor, but huge therapeutic opportunities.
That's fantastic. And what I like about that is you're not only just looking at it, from a science perspective, but you're bringing in other disciplines to try to really take it to the next level. I mean, that's kind of the whole concept of the collaborative. That's the whole concept of what we do as a group from a foundational standpoint. And it's nice to see it on the other side. And I think that's what made this group excited to learn about your grant. Rob, you had a few comments to make here today as well.
I'll start off by saying it is a pleasure to award you a grant from the DIPG Collaborative to the Diamantina Institute for your work.
Thank you.
Your work on
Thank
innovative
you so
models
much.
of brain microenvironment for the treatment of medulloblastoma to the value of $136,561. Congratulations.
Oh, thank you.
Thank you so much.
So what I liked about your grant is that it offers an alternative approach to treating brain cancer, which is not just to focus on the cancer cells themselves, but also on the cells in the tumor microenvironment, the extracellular matrix, the blood vessels, and hypothesizing that the growth of the tumor is cooperative and contributes to treatment resistance. And by targeting this communication, tumor growth may be arrested. So I hope that the interventions that you identify can be applied more broadly to other pediatric brain cancers, and I wish you the very best of success. I'll now hand over to Eleni, who'd like to say a few words as well.
Thank you.
Hello.
Hello.
We are absolutely stoked to be able to award you this grant. It's just been wonderful to be part of this and to watch all of the incredible work that you've been doing, Laura and team. And we also want to make a point and thank you for being part of our Burpees to Beat Brain Cancer Challenge, uploading some of the most incredible videos on our social media with amazing content. It's incredible. We're going to try and put a film together with clips of it all, and we'll send it out when we do.
Okay, sure.
That'll be great. Yeah.
Inspire us all, and we feel really privileged to be able to hand this check over to you. I am now going to hand you over to Kim, who is going to say a few more words.
Thank you.
Hi there, everybody. On behalf of Robert Conadors, we are extremely excited about this grant. I think Rob summed up some of my thoughts too. I've sort of come from the field myself as an individual as well. And what I loved about this was the more holistic approach, looking at not just a single or two components. It really is cutting edge stuff. And I think it really is that next step on that we're looking for, not just in this particular tumour type too, but the lessons learned can be moved over. And that's the bit that hugely excites us. We think it's very cutting edge and, you know, very worthy of getting some funding. So from our point of view, we wish you all the best. We look forward to hearing all of the progress along the way. And I think this, again, because it is at that forefront, it really is offering hope to move the dial forward for all parents out there with children affected by brain tumours in general, but obviously in this specific field as well. So thank you.
Thank you.
Thank you.